One codon is 3 base pairs. And that means that there are 4 times 4 times 4, 64 codons possible. However, we also know that there are 20 amino acids. And therefore there's an excess of 44 codons. And the question is, what do the extra codons do? Now, there are 2 possibilities. One is, of course, that extra codons don't do anything. And the other possibility is that more than one codon can code for an amino acid, can code for the same amino acid. So which of these possibilities is the right one? So, for example, we know UCA codes for serine, and what do UCU, UCG, and UCC do? Evidence that these codons must be coding for other amino acids, in fact, comes from the suppressor mutants, the R2 suppressor mutants that Francis Crick studied. So let's say the wild type message is CAU, 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 and so on, which codes for histidines, and the double mutant is CAU, and then we have an insertion of an A at the 4th position, so that we have ACA, UC, and then the deletion in the suppressor of the A at the 9th position, and then we have UCA, and so on. And if and this results in triplets of the following form: CAU, A, CA, UCU, and then CAU, and so on. Now, the first codon gives us a histidine, the second one gives us a threonine, the third codon is a UCU, and if we assume that extra codons don't do anything, the message would simply stop here, and we would not actually get a reversion of the double mutant back to the wild type state or suppression. Therefore, for the frame shift to be corrected, the extra codons must be doing something. In other words, more than one codon can code for the same amino acid, and this property of the genetic code is known as degeneracy. For example, serine is coded for by 4 different codons: UCA, UCU, UCG, and UCC. Of course, not every amino acid has 4 codons, since that would require 80 codons. It varies between amino acids. However, for most amino acids, more than one codon codes for them. Next, let's discuss how the genetic code was deciphered or cracked. In order to do this, scientists synthesized RNA molecules with a repeating pattern. For example, you could make a synthetic RNA which is repeats of the AGA codon, and then this RNA was translated in vitro. That means it was translated in a test tube, and the resulting polypeptide was analyzed to determine what amino acids were incorporated into that polypeptide. And if you were to do this experiment and found that the polypeptide only contained arginine, you would conclude that AGA codes for the amino acid arginine. Let's do an example. What polypeptide would result from in vitro translation of repeats of CAG? So the message is going to look like the following: CAG, 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 and so on. And what amino acids we get will depend on the reading frame. So the first reading frame would be like this. Reading frame 1. And the second reading frame would be like this. And so on. And the third reading frame would be something like this. So that's reading frame 3. And which reading frame you are in depends on where this repeating pattern starts. So for example, if the message began with CAG, CAG, and so on, we will be in reading frame 1. And then if we look up the genetic code, CAG codes for glycine, and the resulting polypeptide is going to be a polymer of glycines, so that when you digest this to analyze what amino acids have resulted, you will only get one amino acid, which is glycine. So that's situation 1. And situation 2, which is if you are in reading frame 2, is going to be if the first letter happen to be A, so that we have AG, then CAG, 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 and so on. And now, when we are in reading frame 2, then our repeating codons are AGC, and we can look in the table. AGC is serines, and so we will get a polypeptide which is a polymer of serines. And once again, when we analyze this, digest this polypeptide to see what amino acid is made, we would get only serine, and we would infer that AGC codes for serine. And now let's do the third possibility, which is reading frame 3. And this would happen if the first letter happened to be G. So the message is G, CAG, 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 and so on. And looking it up in the genetic code, GCA corresponds to alanine. Therefore, we will get a polymer of alanines. And once again, when we digest this polypeptide, we will we'll have only one amino acid, which is alanine, leading to the conclusion that GCA codes for alanine. And so in this kind of repeating codon messages, you will get one and only one amino acid, but which amino acid will depend on the reading frame of the message. So at a time, you can only get either glycine or serine or alanine, but which one it is will depend on the reading frame, which means where in the repeating pattern the message starts. Next, let's look at another example where we have to determine the amino acids or the polypeptide that would result when we do an in vitro translation of repeats, where we are repeating not 3 letters but just 2 letters, GA. And in this case, the message will look like this: GA, 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 and so on. So if we look at the first reading frame, that's going to give us GAG, then AGA, then GAG, and so on. And if we look at the second reading frame, it's going to give us AGA, GAG, AGA, and so on. And the third reading frame, and let me just label the reading frames here. So red is reading frame 1, green is reading frame 2, and purple is reading frame 3. And so in the third reading frame, we will again get GAG, AGA, GAG, and so on. And we see here that reading frame 3 has the same sequence of amino acids as reading frame 1. And let's see what reading frame 1 or reading frame 3 amino acids are going to be. Well, our codons are alternating between GAG and AGA, and so on. And GAG codes for glutamate, so we'll have glutamate, and AGA codes for arginine, we'll have arginine, and then glutamate, and then arginine. So when this message, when this polypeptide is analyzed, instead of getting a single amino acid, like we got in the previous example of a repeating triplet, we will in fact get a mixture of two amino acids, glutamate and arginine. And now let's see what happens in reading frame 2, where our repeating sequence is AGA, GAG, AGA, and so on. And AGA still codes for arginine, and GAG still codes for glutamate, and we will get an alternating sequence of arginine and glutamate, just like in reading frame 1 and reading frame 3, so that when this polypeptide is digested, we will get a mixture of arginine and glutamate. So in contrast to the situation where the triplet was repeating, where which amino acid you get depended on the reading frame, in this situation where we have a doublet that's repeating, the amino acids you get does not depend on the reading frame. Irrespective of the reading frame, you will always get, in this example, a mixture of glutamate and arginine. However, whereas when you had a repeating triplet, as in the previous example, where you only got one amino acid in the polypeptide, and only one amino acid after you digested the polypeptide, in this case we get a mixture of amino acids after you digest the polypeptide. Having determined which codons code for which amino acids, the last remaining task for decoding or cracking the genetic code was to determine which codons signaled the end of proteins. In other words, what the stop codons were. An insight into stop codons came once again from the T4 phage mutants, in which the sequence of the full length protein was known, and several truncated mutants were found. So perhaps one truncated mutant is like this, another truncated mutant is like this, a third truncated mutant could be like this, another one could be like this. Analysis of the lengths of these truncated proteins and comparison with the sequence of the wild type protein indicated that truncated proteins tended to end where there were very specific amino acids in the full length protein. So glutamate, tyrosine, tryptophan, or serine tended to be in positions that got mutated to a stop codon. And the codon for glutamate is GAG, for tyrosine is UAC or UAU, tryptophan is UGG, and serine is UCG. And these codons, that means, are easy to mutate to a stop codon. And since mutations are rare, it's highly likely that they are in fact just one mutation away from the stop codon. And if you stare at them for a little bit, you realize that if we changed this G to a U, we get UAG for the mutation of the GAG codon for glutamate. And if we change the third position in the codon for tyrosine, the C or the U to a G, we get UAG. And if we change the second position in the codon for tryptophan, the G to an A, then again we get UAG. And if we change the second position in the codon for serine, the C to an A, once more we get UAG. And since all four or five of these codons are one mutation away from the same sequence, UAG, this strongly suggests that the stop codon is UAG, or at least one of the stop codons is UAG. And what's happening in these truncated mutants is that in the normal wild type protein, where there's a glutamate, if you mutate the G, the first G to a U, you get a stop codon and the protein is truncated at that position. The same for tyrosine, tryptophan, and serine. And the reason truncated proteins tend to end at these amino acids is because these codons are just one base pair away from the UAG stop codon, and this UAG stop codon is called the amber codon. And similar types of experiments uncovered two other kinds of codons. One is UGA, this is called the opal codon, and UAA, the ochre stop codon. And through these painstaking experiments and studies, now we know the entire genetic code, which is universal and applies equally from bacteria all the way to human beings. Let's do an example. A double-stranded DNA molecule with the sequence shown here produces a polypeptide that is 4 amino acids long, including the starting methionine but not the stop codon. Determine which one is the template and which one is the coding strand. Label 5' and 3'. What is the sequence of the transcript? What is the sequence of the polypeptide? Now, let's recall that this is a double-stranded DNA molecule, and either strand could be the template, and or conversely, either strand could be the coding strand. And since it's easier to see the RNA sequence in the coding strand because the sequence of RNA is the same as the sequence of the coding strand, except the T's are replaced by U's, we should start by looking for start codons. And start codons are AUGs, methionines, and since this is DNA, we will be looking for ATGs. And here is one ATG, and there is a second ATG going from right to left on the top strand of DNA. So we can see if we start to make a polypeptide at this start codon, does it meet our criteria that the polypeptide should be 4 amino acids long, including the methionine, but not the stop codons. And we're going to be looking for the stop codons UAG, UGA, and UGG. So in DNA, we will be looking for TAG, TGA, and TGG in the coding strand. All right, so we have one codon, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and in none of these sequences do we see, in none of these codons do we see the stop codon. We don't see a TAG or a TGA or a TGG, suggesting that the polypeptide that we're looking for is not starting at this ATG. Now let's try the other one out. So we have ATG, and let's mark this start codon. ATG, TTT, AAG, GCA, and TGA. Now TGA is the stop codon UGA, and therefore we have a stop codon. Now let's check if this polypeptide is going to be 4 amino acids long. We're going to including the starting methionine. So this is methionine, that's 1. This is the second amino acid, third amino acid, and the fourth amino acid. And then you have the stop codon. The stop codon, unlike the start codon, does not add any amino acids. Therefore, this polypeptide is indeed the correct length. And since the top strand is the coding strand, and the bottom strand must be the template strand, and since RNA is going to be transcribed from the right to the left, and the RNA is going to be made 5' to 3', this means that on the template the right is 3' and the left is 5', and the coding strand is anti-parallel to the template strand, so 5' is on the right and 3' is on the left, and our sequence of the transcript is 5' AUG. So that's my start codon or methionine, and once again I'm just reading the sequence of the coding strand and replacing the T's with the U's. UUU, AAG, GCA, and UGA, which is my stop codon, and this ends in 3'. And finally, the polypeptide sequence can be written down by just using the table. The first one we know is methionine, because that's the start codon, and then we have to look up what UUU is, and that's phenylalanine. And then the next one is AAG, and AAG is lysine, and GCA, which is an alanine, and the last codon is the UGA, which is the stop codon, which is not translated into any amino acid, and that just terminates the polypeptide. And here we have the polypeptide of 4 amino acids that would be translated from the transcript that arises from this double-stranded DNA molecule.